Welcome to our review on exchange surface examples. In our previous video we've had a look at the need for these exchange surfaces, and now what we're going to do is actually look at specific examples of where they occur in living things and how they actually work. So the first one we're going to look at is the small intestine in mammals. Hopefully we do remember that the small intestine is the site of absorption into the blood of the nutrients we need. So things like glucose, the amino acids, etc., all getting absorbed into our blood through the small intestine. The way that that's done is either by active transport or diffusion. Now, to help out with this, because the small intestine is to all intents and purposes like a tube that everything passes through, then we obviously need that stuff to travel from the actual intestine into our blood quite quickly. So what we actually have isn't a smooth tube at all. What we find is there are all these tiny finger-like projections called villi on the surface of the small intestine projecting in. And the whole idea behind that is it's going to increase the surface area. Having the villi isn't the only adaptation within our small intestine. We've also got two other key adaptations we should remember. So the first one is that we've only got a single layer of surface cells, and you can see that in the diagram on the left there, that single layer epithelium. Now the reason for that is that it gives us a much shorter diffusion pathway, allowing substances to cross that membrane much faster. The second one is that we've got a really good blood supply. So you can see the actual blood supply in the diagram there. And the idea behind that is to maintain the steep concentration gradient. So as soon as substances have crossed the membrane into the blood, they're carried away to keep us with a steep concentration gradient. The second exchange surface we're going to look at is the gas exchange surface in humans. Hopefully we do know that this is located within our lungs, and the purpose of our lungs is to transfer oxygen into the blood and remove the carbon dioxide from it. And the way that these have been adapted to help let this happen is by having millions of these structures called alveoli. So you can see them on the right there, the little air sacs. And the whole idea behind having all these millions of alveoli is to increase the surface area over which the gas exchange can take place. What we find then is as the air enters the alveolus, then it's actually going to let oxygen move from the alveoli into the blood. And the reason it does that is because there's a higher concentration of oxygen in the alveoli compared to the blood. And carbon dioxide does the opposite. It's going to move from the blood into the alveoli because, again, it's moving down the concentration gradient. The blood's got a higher concentration of carbon dioxide than the alveoli, so it moves from the blood to the alveoli. And you've got my wonderfully drawn in paint diagram on the right, hopefully to illustrate that for you. There are four adaptations of the alveoli that we need to know. First one that we've already mentioned, they've got a large surface area. Second one, they've got very thin walls, only one cell thick again, whole idea, short diffusion pathway. Third one, good blood supply, therefore maintaining that steep concentration gradient. And the last one, the word that everyone loves to hate, the word moist. So they've got this moist surface because that will help dissolve the gases to allow them to transport into our blood. The third example of our exchange surface is the gas exchange surface in fish. Now, when we're talking about fish, the gas exchange surface is actually their gills. So what we see is that as the fish are swimming along quite happily, then the water is entering through their mouth and it passes out through the gills. So if you look at the diagram at the bottom left there, you can see water goes into the mouth and out through the gills. And I've given you a lovely picture of the gills at the top there. As that water is passing out of the gills, then oxygen is going to diffuse from the water into the blood of the fish. And the carbon dioxide diffuses from the blood into the water. In terms of how the gill has been adapted to suit this function, then when we look at the gill, it's actually made from lots of things called gill filaments. And that means we've got a really large surface area again. But in addition to that, those gill filaments are covered by this other structure called lamella, which further increases the surface area. Just like we've seen with our other structures, then we've got lots of blood capillaries associated here. And we've also got a thin surface layer of cells, both of which are obviously designed to increase the rate of diffusion. The lots of blood capillaries means that we're maintaining a steep concentration gradient, and the thin surface layer of cells means that we've got a short diffusion pathway. The final example of a gas exchange surface is in plants. So we're going to have a look at the gas exchange in leaves. Hopefully we do remember that plants need carbon dioxide in order to carry out photosynthesis, and the carbon dioxide is going to come from the air surrounding the plant. So carbon dioxide actually diffuses into these things called air spaces, which you can see on the diagram on the right there, and then it diffuses into the cells from there. There are three key adaptations we should remember to do with our leaves. The first one is if we look at the bottom of the diagram there, then we can see there are these bits called stoma. Now, the stomata are these little pores that are on the underside of the leaf, which allow the carbon dioxide to diffuse in first of all. If we think about the shape of a leaf, then we know they're quite flat, and the whole idea behind that is to increase the area of exchange surface. And the last one, those air spaces, are going to increase the internal surface area for diffusion to happen over. Hopefully at the end of this video you can recall examples of different exchange surfaces in both humans and in plants, and you can also describe how they have been adapted to improve the rates of diffusion.